16th, maybe in evening after 7, a quiz can be done. Also, like audit and withdrawal window has not yet opened, and even if it opens, it will remain open for 7 days. So I don't know why people are very angry, and they are sending me WhatsApp message and what, like, why did you ask for this? I don't understand such things. And not in this course, in one another course also, like, people are like against me, like, what to do. I cannot say anything, although I'm okay with anything.
Yeah, I think it's all right. Look, I think if you feel, if all of you still feel 12th is early, then we can even like I said, we can have it on 14th also. In 12th, we can have our regular class. For me, it doesn't matter. But then our next class is going to be on 14th. So we can have the quiz on 14th also. So those of you who are present, let me take your consensus on this. If you want more time, you can study over the weekend a little bit more. Then on 14th also, we can have our quiz in the Monday class. Okay.
Yes,
So
madam.
again, the same thing, that first 9:30 to 10:30, we will have our regular class, and then 10:30 to 11, we will start the quiz. So I think, Vivek, is that all right? I see you have logged in. Vivek Sehra. Abhishek.
Yes, ma'am. Yes, ma'am.
So I'll send a note through MS Teams to everybody. So let's keep it in that case on 14th, and 12th we will have our regular class. And the only thing, before I start our regular lecture today, is that you have to make a presentation on your term paper. Now, our semester, our last date for the semester is only 1st April. So I was thinking that in the class of 28th March and 31st March, I'm going to schedule the presentation of the term papers. So between now, so you have almost 18 days in which, the term paper, you can submit the term paper, the written part later, but the presentation which is going to be on the term paper is going to be scheduled on 28th and 31st. So please take note of that. I will also circulate. So, you give yourself mentally whatever topic you are going to work on. You start to prepare, and, you will just get 6 minutes. We have so many people who are going to make a presentation. So I will just have 6 minutes for everybody. And if then there is a spillover, we can, we can manage another date. So with this, let's start with the regular lecture today. Okay, so essentially we started to talk about phytoremediation, and this is where we were last time, that we are looking at case studies whereby trees like ficus, for example, there is just a picture of that given in this slide, whether large trees like ficus can be used for remediation of metals in a soil which is contaminated with these metal ions. Okay, so is this slide visible to everybody?
Yes, ma'am.
And if you have any questions in between, please interrupt me. So the overall layout, in order to understand what the experiment is going to deal with, is that what you are looking at here, for example, is a land where you have soil which is contaminated. The trees are going to be now planted, which will be at, very small stage. The trees are going to be transplanted into this soil, which covers an area of about 400 square meters. And because of its capacity to take up these heavy metal ions, the metal ions which were studied were cadmium, copper, lead, and mercury. All of these are heavy metals, and they are contaminants. So it is going to take up the plant, because of its ability to take up the heavy metal ions and transport them in its system from the roots to the shoot, for example. Many of these heavy metals are taken up. So this part of the experiment was carried out for about 3 months, and after which these plants were uprooted after 3 months. And then, of course, the roots were taken from they were sampled, the shoots were also sampled, and many of them were now transplanted into a fresh and a clean soil, okay, at a site which is other than the contaminated soil. So what is done now is that it was also measured if the metals which have been now accumulated inside the shoot, inside the root, for example, are going to be coming out into the bulk soil. Okay, because obviously through diffusion, through efflux transporters, and because the concentration of these is very low in the clean soil, then are these heavy metals going to be coming out of the plant roots? So therefore, you measure, for example, what is the metal which has come out into the clean soil. And for this, two types of soils were checked. One is the bulk soil, that is majority of the soil which is around. The second is the rhizosphere, which is the soil which makes a very intimate contact with the root. So it is the soil which actually attaches itself to the root. That's called as the rhizosphere. So this is the overall layout of the work. Now, experimental details are given here. It is only for your purpose. I will only read out one box, but otherwise you please, it is, whatever I just said a couple of minutes ago is explained in this layout. That uniform, so these are the plants which have nearly similar morphology in terms of their height and diameter at the trunk, or the crown diameter, that is, how much it is at the top. So nearly plants with similar morphology are taken and they are planted at a certain density. And then 4 samples are collected immediately, which serve as a control, because at time 0, you want to know what is the level of these metal ions in the root or in the shoot or in other parts of the plant. And then it is allowed to grow, for example, as it says in the third box, that the remaining plants are allowed to go for 3 months. Okay, and then after 3 months, they were uprooted, they are cleaned, and parts are again taken, the root, the stem, and the leaves, and the metals are sampled in these parts, and eventually the plants are washed and they are transplanted again and allowed to grow for 3 months. So this is exactly the way the experiment is planned out. So what is being measured here quantitatively? There are 3 things which are measured here. Number 1, what is called as a bioaccumulation factor. Okay, what does it mean? It means it is the metal content in the plant tissues. That means how much of metal are you looking at, milligram per kilogram of plant tissue. And this tissue is either the root tissue or it is the leaf tissue. Okay, divided by the metal content in the corresponding soil. So in other words, how much of metal has really accumulated now in both roots as well as your leaves, divided by the total metal content in the corresponding soil, which is indicative of the metal absorption capacity of the plant. Okay, and the second thing which has been measured is what is called as a translocation factor, which means the metal content in the plant aerial tissue. That is, how much of the metal do you really detect in the leaf divided by the metal content in the plant root, which is indicative of the metal transportation. Because, first the metals have come in through the roots, and then how much it is really transported to the leaf. That is an indication of metal transportation. And finally, you talk about the decontamination capacity. That is the total metal which is extracted, okay, and which is the metal content in the plants multiplied by the biomass yield of the plant. Okay, so metal content could be, for example, you have so many milligram of metal, for example, per gram of tissue. And then in 3 months' time, the plants have also grown. So you are going to normalize it with respect to the total biomass which you have accumulated. So make sure that you understand what the term means. And this indicates the decontamination capacity in the experiment. So are these 3 terms clear to everybody? Yes or no? Yeah.
In the first point, where you mentioned that metal contents in plant tissue, so are these any random tissue from the plant?
Well, as I said, in the experimental protocol, it says that the roots are collected separately, the stem is collected separately, and the leaves are collected separately. Okay, so these are the only 3 tissue types which have been taken. That is the soil, the stem of the plant, and also the leaf of the plant. So all of it is really added up, and then it is divided by the metal content in the corresponding soil. That is, you know, when you started, for example, there was milligram per kilogram of soil. Similarly, now you have milligram per kilogram of plant tissues. So that is an indication of the metal absorption capacity. So is this point clear to everybody?
Yes.
Okay. So now in the total area, you know, you have like 400 square meter, which is planted with these trees. So initially, what was the metal content? Now, the four pictures which you see here, they reflect the metal status of cadmium, copper, for example, mercury, and lead. Okay, and there are three different symbols which are used here to mark the areas. So the one which you see here, for example, is the one which contains less than 0.3 milligram of cadmium per kilogram of soil. And this zone is called as the relatively clean zone. Okay, because remember, you know, the plants are also going to be transplanted over the entire area, and the distribution of the metal is not exactly the same in all over this area where you have planted your trees. So you need to know, even when you are carrying out when you are planting your plants, you need to know what was the initial initial metal content in the soil. It is not uniform, as you see here, for example. So these are the areas which will have less than 0.3 milligram per kilogram of soil. Okay, so they were called as relatively clean soil. And then you have like these hashed lines, which indicates that the cadmium which was present is between 0.3 to 0.6 milligram per kilogram of soil. So this is what you begin with on the land. And similarly, what you see this part, for example, here is more than 0.6 milligram of cadmium was present per kilogram of soil. Okay, so try to understand what it means. And so in other words, you know, this is so that you can call now something which is clean, or which is moderately contaminated, or which is heavily contaminated. And at the same time, the numbers which you are seeing here is that everything, whatever you have, adds up to 100. So 43% of the total area which you are looking at, for instance, is moderately contaminated. 18% of the total area is heavily contaminated with cadmium. And of course, the remaining area is clean. So 100, you know, you add these two numbers and you subtract them from 100. That will give you what is the percent area here. All right, so I hope these numbers make sense and you are able to interpret them properly. Okay, similarly, for instance, if we look at mercury, so you see that the area is very heavily contaminated with mercury. 64% of the area is heavily contaminated, and that also means that it has more than 0.7 milligram of mercury per kilogram of soil. There is very little clean soil, and then there is also the moderately clean, which is 27% of the total area, if you are only examining the mercury content. Okay, so that is the way the data needs to be interpreted here. So the data for copper is given, the data for lead is also given. So now let me just show you, for example, because you know, you need to look at these tables and you need to understand, because in your quiz, for example, I could just give you one table, I could give different data to different people in their papers. Okay, and then you could see, for example, what is the information which you are supposed to be extracting. Okay, so let's take the case of cadmium. So for cadmium, the clean, the area, you know, what you define as clean, which has a certain level of cadmium, there the samples are 39. That is 39 plants were uprooted from there and they were sampled. And cadmium concentration was measured in them. What was the range of the soil metal content here in this particular area? Okay, so what you are looking at here, for example, because initially, you know, you are not measuring the metal content in the plants, because that is your time zero. But what you are measuring is the metal content in the soil. Okay, so what is associated at time zero with these plants ranged from 0.04 to 0.29. And average was so much. The root biomass, so if you have got 39 of these, all right, then you have, you know, if you have 39 of these plants, the total weight of this is 4.99 gram. The shoot biomass, that is the leaves, etc., is 7.44. The whole biomass is only a sum of root plus the shoot part. In the heavily polluted soil, for example, you have, 18 plants were sampled, and the range, because obviously, there is a little bit of a variation. The number is not constant, and that is the reason why a certain range is given here. Because you are looking at the soil which surrounds 18 of these plants. It's not exactly constant. In one plant, you found 0.62 milligram of soil. In the 16th plant, you found 0.71. So this is the range which you are dealing with at time 0. Okay, so make sure that you are able to understand. The same data is also given for copper, mercury, and lead. So make sure that you are able to understand what does the data here refer to. Okay, now another point which I want to emphasize here is that, so is this clear to everybody?
Ma'am, why
here
they have taken a different number of plants? It would have been better to compare if the number of plants would have been like same.
Yeah. I'll tell you, the reason is that, number one, if you look at the distribution of the clean, the polluted, and the heavily polluted areas in the previous slide, for example, look at cadmium. Okay, now you see 18% is only heavily contaminated, 43% moderately contaminated, and this is slightly contaminated because the density of the plants, they have kept the same. Okay, if you wanted to keep the sample number the same, then you would have to plant a lot more trees in this. Okay, so if you look at, for example, in this slide, okay, so the total density is 2,500 plants per hectare of land in order to not allow any plant-plant interaction to occur, because the density they wanted to keep the same.
Yes, ma'am, understood.
Okay, all right. Because otherwise the plants will compete with each other, and there are so many other phenomena which are occurring, so that you did not want to do. Okay, that is the reason why your samples here are quite different. So make sure that you understand, what these numbers are being referred to.
Ma'am,
So,
number of plants, right?
yeah, please repeat that again. What is the question?
N, which is written over here, let's say for cadmium, it is written N is equals to 39. So this
Yeah,
means number of plants that are there.
absolutely,
So
absolutely. So
they are
39
comparing
plants. Yeah,
out of 39 were at like time t is equals to 0, which were initially clean.
yeah.
And then 43 came out to be moderately polluted and 18 came out to be heavily polluted, right?
Heavily polluted, very true, because it is from heavily polluted soil, and that is why when you look at the soil metal content also, it is much more compared to what you see in the other two category.
Yes.
Okay, that is to be expected, because if you are sampling from the cleaner environment like copper, for example, it's only the range was 15.2 to 49.8, but then what you call as moderately or just polluted, they have used the term there, it is more than what you have here. What you call as heavily polluted is now much more. Okay, so that's what these numbers refer to. So is that clear now?
Yes, ma'am.
Okay. Let's move to this particular figure. So what is being represented here on the y-axis is, for example, the total amount of metal which has accumulated in the plant. Okay, and that is, for example, this figure here represents what is the total amount of cadmium which has accumulated in the plant. This cadmium is measured in both root as well as in the shoot area. What you are measuring in the root is shown as these squares, and what is shown as inverted triangle is shown, for example, in the shoot. What is on the x-axis is as the soil cadmium concentration changes. Because remember, in the last sample here, it is not constant. The concentration of the soil, of the metal content which is in the soil is not the same. It depends on from where the tree has been sampled now. Okay, and that is the reason why in this figure there is a variability. So you want to know whether it is a linear function of this. Does it continue to increase for all metals, or does it really plateau off for certain metals? Because that is going to reflect something else which is happening. So for cadmium, if we look at the data, so as the soil cadmium concentration increases, which is the milligram of cadmium per kilogram of soil, as it increases, what you see that the total amount of cadmium which you measure in the root, for example, also more or less increases linearly up to a certain level. Then you see that in this particular zone, it starts to plateau off. So there is a saturation which you are achieving. Okay, but up to a certain time, in other words, that as cadmium concentration increases in the soil, the plant's ability to accumulate this also increases up to a certain level. After that, it plateaus off. The same situation you also see in the shoot. Okay, that means there is adequate transportation. In other words, it is an example of phytoextraction, not phytostabilization. Because phytoextraction means that whatever, obviously, shoot has never come in contact with the metal. It is only the roots which have directly come into contact with the metal, but then they are able to transport cadmium along the xylem and they are able to leave it behind in the shoot. Shoot means the aerial parts, which are like your branches or which are going to be your leaves, etc. So when you measure, again, the metal content continues to increase up to a certain point, and after that, you see, after 0.5, for example, it kind of plateaus off. So it has already reached its saturation. So in this case, we see that cadmium is linearly taken up to a certain point, both in root as well as in shoot. So you are doing phytoextraction of cadmium, not phytostabilization. Is that point clear to everybody?
Yes, ma'am. Yes, ma'am.
Okay, so now if we see what happens to copper, for example, so you see now in copper that the total amount of copper which is accumulated is much more, and that goes between 10 to nearly, I think this is like 20, 30, 40, 50, up to 60 milligram. So it continues to accumulate and then suddenly it plateaus off at this particular level. Okay, but when you compare, when you look at the data with respect to shoot, you see that there is not as much which is taken up in the shoot. So it is only a question of phytostabilization. It's not a question of phytoextraction, because copper doesn't get transported as much into the leaves as cadmium does. So is that clear?
Yes, ma'am.
Yes, ma'am. It is clear, but when we are saying this, that in soil, concentration of this cadmium, for example, is 0.1 mg per kg. On the other hand, in plant, it is something like much higher concentration is becoming is in the plant tissue. So
Exactly.
like from where they are like till the point the roots are spread in the soil, they can get, but like how they are able to extract more cadmium than the like they can reach.
Well, I think the first thing which you need to note down is that for cadmium, number one, okay, the total amount which you are dealing with is much less. That is a valid point. But then, these are the actual numbers which are from the soil, because nothing has been added from outside except the electronic wastes, like the batteries or the e-wastes are just dumped into that area. Then you take a stock of how much of cadmium really comes out of these electronic wastes, how much of copper comes, how much of lead comes, and how much of mercury comes, because this is the real life situation. You don't want to change these numbers. This is what is exactly present in the soil. Okay, that's it. So therefore, in the soil, then the amount of copper also varies from 20 milligram per kilogram to 150 milligram per kilogram of soil. But the cadmium, the leachate of cadmium is not as much as it is for this or as it is for lead. Okay. However, it may not be entirely due to it may not be entirely due to the kind of metal ion there is, because copper, for example, is also transported very quickly by there are certain copper transporters inside the cell, because copper is also an essential metal for all the cytochromes. So its capability of taking up copper is much more compared to that of cadmium. And for copper, there are specific transporters, whereas for cadmium, there may not be specific transporters which are located.
Yes,
So
ma'am.
exactly. So under the given conditions, because these are all the real life numbers, it's not that you have taken the soil and you have now spiked it, imagining that this is what is going to be. But these are actually, these are the real life sites where there is electronic dumping, the wastes are dumped, and then this is the concentration which is found already in the soil. So your data is also based on the real life scenario.
Ma'am, you said that for copper there is transporters, but for cadmium there are no transporters. But when we see like plant Cu, ratio of plant Cu is to soil Cu, it is higher for cadmium. On the other hand, it is very less for copper. So like it will be like 5 for cadmium and half, I guess, very less for copper.
No, you see, but if you see the total amount which is accumulated is also, if you see, that this is like going from 10 to 100 now on this paper, whereas this is a linear scale and this is only 2 milligram per kilogram.
Yes, ma'am, that is there.
All right. So please, make sure that you also see what is on the y-axis. This is a linear scale and this is a log scale here. All right. Of course, scale for mercury is even lesser. You see that there is not too much of mercury in there, but nevertheless, mercury is found, even if it is to a small extent, but mercury is taken up inside. And in the soil, if you look at the mercury, this is what the spectrum looks like. And again, you do see a lot of accumulation of this in the root, whereas in the shoot there is very small accumulation, which is almost, if you look at this, is 40-fold lesser compared to what you see here. So that means again there is a phytostabilization. There is no phytoextraction of this metal, of mercury.
Yes, ma'am.
All right, so these numbers you just need to understand now. And what's important here is that, why do you really measure, for example, how much of this is in the shoot, etc. Because photosynthetic parts like leaves, for example, okay, or some of those green branches are also the sites where the plants are going to fix up food. So photosynthetic organs are extremely important, because plants don't take up carbohydrates from outside. They make their own food by fixing up carbon dioxide with the help of the leaf, etc. And that's the reason why there is so much of stress on what is the concentration of metal, like it says here. Concentration of metal in the non-photosynthetic organ is very, very important. Okay, so the average transportation factor of cadmium is 0.89. So if you were to look at, for example, what is the average number here, it is 0.89, and the range goes from 0.61 to 1.38. That is how much of it is really transported. So what do you have in the shoot vis-a-vis what do you have in the root? Okay, and it also indicates that there is a high bioavailability of cadmium. By bioavailability, what do we mean? That quite a lot of cadmium is taken up from the soil and it is also made available. It is transported through the system. Okay, so that's what these numbers mean. And it also indicates that ficus is a suitable candidate for cadmium phytoextraction, because you have seen yourself that unlike the data what you get with copper or with lead or with mercury, the trend of what goes into the leaf follows the same pattern as that in the root. So as the roots take up, they are also mobilizing these metal ions to go upward into the shoot.
Yes, ma'am.
So now let's look at this particular figure here. It's from the same paper. Now, you know, all of these, for example, the metals, the idea is to be able to explain that in the root, you know, where exactly are these metals getting deposited and what is the chemistry behind it? Are there any, is it simply by diffusion, or are there special molecules which are localized within the root tissues which are able to absorb much of these metal ions? Okay, so in order to understand, here is in this picture, you see there is a cross section of a root here. So imagine that you have taken a root and then you just take a cross section, take a section of it and examine it under the microscope. So what you are going to look at, for example, is the outermost layer which you see in the gray, okay, and that is called as the epidermis, which is what is labeled here. If you see here, so epidermis here is the outermost layer of cells. Okay, and then the cells which you see in the yellow, for example, okay, that is called as the cortex. And it is, if you can read it here, this is cortex. So your arrow points to this section. So all the cells which are shown in the yellow is called as the cortex. And finally, this particular region, which extends from this dark gray to this, okay, so inward, this area is called as the stele. Okay, so I hope it is clear. So what we are trying to look at now is the concentration, for example, of these metals. How much of metal, for example, if you look at your y-axis, EPI divided by CAS, cadmium, that is how much of cadmium is present in the epidermis divided by cortex and stele. So in other words, if there is a lot more of the metal ion which is now in the epidermis, you will expect to see this ratio to be very, very high. But the lower the number comes out to be, that means now there is, of course, the number is always, you know, it is in whole numbers. You will see, for example, here, if you look at this ratio for cadmium, it extends from 5 to 16. If you look at copper, for example, the ratio goes between, for example, 2.5 all the way up to 8. So it is in whole numbers. That means the concentration of the metal is always more in epidermis compared to that in cortex and stele. But how much more? And what happens as a function of increase in the soil cadmium concentration? This is what the figure is trying to tell you. All right, so please take a minute and try to understand what this data is trying to tell you.
Yes, ma'am. But the as we know that this cadmium can go upwards using this xylem tissue. So why they are being accumulated in epidermis? Epidermis is the outermost part of this cross section.
Exactly.
So.
So the reason is that, you know, there are, I think I have this thing here in the last explanation that even in the epidermis, okay, there are certain chemicals which are present. Okay, now let me read out the last point to you. The first of all, you know, we will come to that point later, but first let's try to look at the general trend in all the four figures. So you see, as the soil cadmium concentration increases, there is a decrease in the ratio of what is in the epidermis divided by what is in cortex and stele. Okay, that means at lower concentration of cadmium, everything gets kind of accumulated in the epidermis. So it acts as a buffer. The outer cell layer, which you see, for example, in the root here, the outermost cell layer tends to act as a buffer because there are carbohydrates. There are glycoproteins in the epidermis which can bind to this. Okay, that is a point which is written here also. Abundant carbohydrate compounds like glycoproteins in the epidermis can bind and they can absorb the metals at a lower concentration. Okay, but as the concentration increases, All right. So what you see, therefore, is that there is only a certain capacity of all these of these carbohydrates which are in the epidermis. And once they attain saturation, then these metal ions, they start to go into the cortex and the steel region also, as a result of which the ratio also falls low. Because initially the epidermis serves as a buffer, it tends to absorb all the metal which is coming in at a low concentration. It can do that, but as the concentration, so this is the kind of range which they have in all the samples. Okay, but then overall you see that how it distributes. So as the metal concentration increases, all right, you also see that the ratio starts to go low because it has attained saturation.
Yes, ma'am. One thing is this ratio EPI upon CAS here we have. So is this ratio of concentration or total mass
No,
of this sieve?
it is just the concentration. Let me read out the second point to you. Okay, so here it says cadmium, copper, mercury, and lead concentration ratio. So concentration means milligram per kilogram of tissue.
Yes, ma'am. Understood.
Milligram per kilogram total tissue. So, when you weigh out all the roots, etc., you have cut it into small pieces or whatever, then you are going to weigh that and you will find out what is the weight of that. Okay, so it is all in concentration, which is going to be milligram per kilogram of the root tissue. So these are absolute numbers because you are looking at the ratios here. And so in general, this is what the trend is like. Okay, and that is what is explained here, that first of all, more metals they have accumulated in the epidermis, you see, and at lower concentration of the metal, almost all the metal resides within the epidermis. Okay, and the ratio, what is the range of the ratio is given here. For example, for cadmium, it is from 5.25 to 15.67. Where do I get that number from? Go back to the y-axis and you see for cadmium, the ratio varies from somewhere from 5.25 all the way up to 15.67. So when you are trying to look at these numbers, they are just taken from the y-axis for all the four metals. Number 3, the decreasing ratio as a function of increasing metal indicates that the epidermis, it could be saturated by increased external metal, has acted as a buffer for metals. And there are carbohydrate compounds like glycoproteins in the epidermis which can bind and they can absorb the metals, and therefore they subsequently retard the migration of toxin to metabolic organ. By that what we mean is that metabolic organs are your leaves, etc. So they kind of stabilize all that there. Okay, so is this clear to everybody?
Ma'am,
So
this structure is for root only. This structure is not for like the whole plant cell.
no, no,
When
no,
we are talking about epidermis, then it is for only the what is
only
called
for
root.
roots.
Okay,
Absolutely, it's
so
only for
now
roots.
I understood. Okay.
Okay, because so what you have measured here is, because why so much emphasis there? Because this is the only part of the plant which is making a direct contact with the metal ion. Leaves are not making a direct contact. So whatever ends up in the leaf is because of phytoextraction and transport of the metal. So roots are the only ones which are making a direct contact with the metal. So you want to know what part of the root is it really? If it is the outermost part, what is the biochemical reason for that? Well, it is because it is rich in glycoprotein. For copper particularly, they have found, as is written here, that copper taken up into the cells is tightly bound to homogalactouranon in the cell wall pectin. Okay, so there is a particular component which is present in the cell wall which specifically binds to copper. Okay, so in other words, you're also trying to provide a biochemical explanation for the same. So let's move to the next slide now. So please look at it very, very carefully. So this is now in this table. I think this I have already discussed with you. So in this table, what we are trying to look at is the metal concentration of the soil and the plant tissues before and after transplantation. Okay, let's take the case of cadmium. And this is, the plants were cultivated on the clean soil, or they had come from the polluted, or they had come from the heavily polluted soil. All right, so let me give you this example. So metal content before the transplantation. So when after growing it on heavily polluted soil, you measured what is the cadmium concentration. It was found to be, 8.27 microgram per kilogram of the plant tissue. Okay, and metal content before transplantation in the shoot was found to be 12, but after transplantation the number has gone down to 4.91. Okay, so before transplantation it was 8.27. After you transplanted it into the clean soil, it became 4.91. That means some of the metal which the plant had accumulated in its root, it has gone out into the rhizosphere or into the bulk soil after transplantation. Is that clear to everybody?
Yes, ma'am.
Okay. Similarly, before transplantation, there was 7.29 microgram. After transplantation, it has come down to 5.13. Okay, so the plants have actually acted as carriers of various metal ions. So from the polluted soil, they carried forward certain metals, and now they have released it into the cleaner areas. If you look at what is the scenario with respect to shoot, now there is also mobilization. In the shoot, for example, before transplantation, it is 4.67. After transplantation, it is 5.75 for cadmium. So it is 10.25. After transplantation, 22.5. So in the shoots also after transplantation, a lot of this metal got accumulated now. So in the roots, it has lessened. Either it has gone into rhizosphere or it has moved upward into the shoot. Okay, now let's take the scenario with respect to copper. In copper, before transplantation, it is 104 and it is 86. But if you look at what happens to shoot, it was 18.0 before, and after transplantation, it is 18.9. Okay, so not much phytoextraction, but 25.9 and 36.1, 29.5 and 56.9. So at this stage, a lot of this has also migrated into shoot after transplantation. Okay, with respect to mercury, if you look at the numbers, the metal content before transplantation, after transplantation is nearly the same. Okay, so there's not much difference. So if a statistical test was performed, these numbers were found not to be significant. Okay, so make sure you learn how to interpret this data. Okay, and what is important is now this is the soil metal content after transplantation. Okay, so in the bulk soil, if we look at, for example, the cadmium, or let's look at the plant which came from after growing on the heavily polluted area in the bulk soil, which is majority of the soil which is a little far away from the roots. The metal content is 0.08 milligram per kilogram of soil, but in the rhizosphere it is a lot more. So that means much of the metal sticks to the rhizosphere. It has not moved into the bulk soil. Is this clear to everybody, this point? So please look at these numbers very carefully, because it tells you, as I had shown you in this picture, okay, this is the difference. The bulk soil is what is really far away from the plant roots, but rhizosphere is where it makes an intimate contact with the roots. So we are trying to measure what is the amount of metal which is in the bulk soil and in the rhizosphere. I hope that point is clear to everybody.
Yes, ma'am.
So this soil metal content after transplantation measures what is the metal content in the bulk soil and what is in the rhizosphere in almost all the cases, except, maybe like for cadmium, you see that there is a tremendous difference. So it has been able to release cadmium. If, for example, if you look at, for example, you look at copper clean, there is not much of a difference here. For mercury, of course, if it comes from little polluted or heavily polluted soil, it's certainly statistically significant. And similarly for lead also, this is the difference. Okay, so what is the finding from this, which was the actual case study, that ficus is a very good plant for cadmium phytoextraction. And make sure you try to understand the difference between phytoextraction and why we are saying that copper, mercury, and lead were only phytostabilized. They were not phytoextracted. And various metals are transferred from roots to shoots and the rhizosphere at different levels after transplantation. Cadmium changed significantly in the bulk soil. After transplantation, the highest concentration of the metal was still far below their corresponding safe threshold. So even when you measure what is the concentration in the bulk soil, okay, that number was found to be still much lower than what are the permissible levels of metal ions in the soil. So therefore, there is not a heavy contamination of the clean soil after the transplantation took place. Now, none of the metals was detected in the leaves, indicating extremely low environmental risk to cultivate the greening plant from the polluted soil on the clean soil. Okay, so the concentration of the metal did not build too much. And this strategy indicated that fertile lands can be recovered after such experiments. So it is possible that, soils which have been cut, because, today, of course, the scenario is very different because people have become quite aware of what should be done with the e-waste and they are not dumping them. But 20 years ago, it was not the case. People were just dumping them without paying any attention to what was happening. Okay, so in other words, those lands which are already not accessible to agriculture or for other purposes can still be recovered by planting trees on them. Okay, so is this point clear to everybody?
Yes, ma'am.
All right. So very quickly, I will do another case study with you, which is with regards to the treatment of textile waste waters. All right. So here is in Maharashtra and in several other parts of Maharashtra only, and because of the presence of National Environmental Engineering Research Institute, NERI, which is a CSIR lab, they took quite a lead in phytoremediation. Okay. And along in Maharashtra, Gujarat, there is also a lot of textile activity which is going on. Coloring industry is huge in Gujarat and along the banks of Maharashtra, because obviously they can discharge their effluent into the water bodies, into the oceans, etc., or in water bodies which are very close to the ocean from where they think it can be released into the ocean. But today they understand that it is a problem. Okay, so this particular experiment has been set up in one of these in one of these very close to the industry, close to the textile industry. And it is from a paper which came out in 2018. Okay, so what you see here is the real picture. Okay, so you see this is called as a ridge and this is called as a furrow. So the effluent, the common effluent treatment plant on the site, it permits primarily treated wastewater through the furrows of this wetland. Okay, so there is a CTP, which is a common treatment effluent plant, and what it does is that all the effluent is now released into this particular, it's a huge area of land where these ornamental plants have been planted. Okay, so what you see here is a ridge and what you see here is a furrow. So that is like a gap, where this effluent can just come out and it can be there. Okay, so if we look at the schematic view, this is only one furrow which you are looking at. Okay, so this experiment was actually conducted over a large area of land, which was in length about 91.4 meters. So it's a land which is about 1/10 of a kilometer. Okay, and 1 meter is actually the width. So here you have approximately, so in terms of the length, it's going to be large, and this is done only over a period, only over a length of 1 meter. Okay, so the construction of such an area is also called as a wetland. construction. So what is done now is that, you have like ridges which are constructed, which could be gravel, which could be snow, which could be, sand or whatever. As you see here, this is the raised part, which you see here. So soil and other things, etc. So this is what it looks like if you look at the cross section of this. Okay, this is called as a wetland. And then in between the furrows, which you see here, all right, is approximately 0.6 meters. Right here. Okay, so this ridge is about 1 meter and this is about 0.6 meter. So this way it is conducted over a very large area which spreads over a total area of 91 meters. And then on these ridges there are plants which have been planted. For example, here you have daguerreotypes, and here aster, which is an ornamental plant, and then you have some other plant here, then you have some other plant which have been planted up here. Okay, so in this experiment, you are trying to see that whether putting these plants here will take away the color which appears in this textile effluent, and what happens to the soil which now comes into contact with this effluent. Okay, so is this layout clear to everybody?
Yes, ma'am.
All right, so in between, you see, this soil is now going to be watered with the textile wastewater. And of course, there is textile wastewater, which is also in these furrows, which are in this wetland. So what is the rationale of this study? What is the logic? Is that, in such kind of experiments, the plants which have been selected are called as the high rate transpiration system. In other words, these plants, they have a very high rate of transpiration. You know what is transpiration? Is that leaves, the plants, they take up a lot of water from the soil. However, a lot of water is also evaporated through small openings, which are called as stomata, which are on the underside of the leaf. Do you remember what the stomata are?
Yes, ma'am. Yes.
Okay, these are openings which are on the lower side, because otherwise water will keep on accumulating inside the plant and it doesn't need so much water. Plus, water is also made as a byproduct of metabolic activities. When you break down, when they use starch, when they do glycolytic pathway, when they do the TCA cycle pathway, they make a lot of water. So they need to get rid of water, and that they achieve through stomata. Okay, so these particular plant, they have a very high rate of transpiration. That is, they take up a lot of water. If you were to quantitate it, that is liters per hour of water which is taken up, and liters per per hour which is evaporated is very, very high for these plants. And these are called as the high rate transpiration systems. Okay, and these were plants like Azadirachta, Eucalyptus, Dalbergia, Acacia, etc. Okay, so they have the potential to absorb huge amounts of water. And that's the reason why they have been selected, so that, along with the water, and if you plant them in areas which are contaminated with textile dyes, then obviously many of these dyes will also enter into the plants. So they are taking up anyway large amounts of water. Along with that, the textile dyes will also go inside the plant. Okay, and this high rate transpiration is achieved through stomata, which works like a biopump, and several garden ornamental flower plants, etc. They were also observed as potential candidate for degradation of textile dyes. I will tell you why, because in phytoremediation, one of the mechanisms was that plants also produce a lot of these dye degrading enzymes like lacases, azoreductases, etc. in their roots, as a result of which they can also serve as a very good system for degradation of textile dyes and for cleaning up those waters. Okay, so is this clear to everybody?
Yes, ma'am. But in fact, when we are seeing this transpiration, so along the water goes from roots to shoot, and after all to leaves, and then it is evaporated. So in that way, as the concentration, as the amount of water, whatever the amount of water goes to leaves, they all take this dyes also to
No,
till
no.
the leaves.
No,
And
the dyes are left behind. The dyes are, as you will see, that the dyes will only appear, they can appear in the leaves, but they also appear a lot more in the roots. So they are able to retain all of these, or they are simply able to break down the dyes. I will show you in the subsequent slides. Okay, because there is also phytoremediation which is occurring, not just phytoextraction or simply phytostabilization. Okay, but there is actually phytodegradation. So that's what we will see in the coming few slides. Okay, so in this particular experiment, there were 4 ornamental plants, because obviously the idea was that while you are doing all this phytoremediation, why not keep flowering plants also, because it adds an aesthetic beauty to that place, and at the same time you are also able to get the job done. Okay, so now this is the experimental outlay which I have just made from the paper, that there were 2,000 plants which were independently planted on these ridge beds, like you see. So what are the ridge beds? These are the ridge beds. So these are the plants which have been put there. Okay, 2,000, and they are watered initially with normal water for the first 30 days. After that, half, that is 1,000 plants, are nourished with the normal water, which serve as a control, and the remaining 1,000 plants were now treated with the real textile waste water for the remaining 30 days of the experiment. Okay, then soil is collected from, so you are going to remove some plants from there, and the soil which is just adherent to their rhizosphere is taken both from the normal water treated plant as well as the textile water treated plant. So the soil is collected, the soil samples were dissolved in HPLC grade methanol, filtered, and then you measure what is the amount of dye, and then they have also taken the root samples and they have tried to mash it and measure the enzyme activities like laccases or certain other these enzymes which if the plants are making, you should be able to measure them in the presence of the textile dye. So now let's look at the results first of all. All right, so on the y-axis, you have the residual dye concentration. So in this stream, for example, what was the dye concentration initially and what is the dye concentration after a period of 30 days when you have transplanted these ornamental plants. Okay, so what you see right here is that, so this is a control where no plant was planted on a ridge. So one of the ridges also serves as a control where there is no plantation. Okay, if there is no plant on that ridge, then you see that the residual dye concentration gradually increases as a function of time. Why? Because textile industry is constantly releasing the effluent. So over a period of time, the dye concentration builds up to a large extent in this water body. In these, in that ridge, for example, where there were no plants. That is to be expected, it cannot remain constant because from the common treatment plant, continuously this effluent is coming in. Now, what happens to those ridges where you had plants which were planted? So what you find here, for example, the open circle here refers to one plant, the triangle refers to the other plant, or soil control. Okay, so this is something which just serves as a control where there is absolutely no dye also in that. So in those water bodies where there is no dye which is coming, obviously the concentration of the dye is going to be 0. So this is the other control which you have. But what is important to note is that on the 4 ridges where plants were put, okay, there is a significant reduction in the total amount of dye. Okay, so what goes out in these streams after a period of 30 days, gradually, when you measure what is the dye concentration, you find that the dye concentration is much less. If there was no plants, then it would be like 1200 milligram, 1300 milligram, but now it is approximately, let's say 450 milligram of dye. And this particular plant was the most effective one in terms of reduction in the total color content over a period of 30 days. Okay, so is this clear to everybody?
Yes, ma'am.
Okay.
Yes.
All right. So now what they have done is that from both the control. Okay, so the control lane was the one where the plants were treated or they were watered with normal water. Then you had a test series in which 1,000 plants were now watered with the textile dyes. Okay, and the number of soil bacteria. So this is from the rhizosphere. Rhizosphere means that the soil which was making a very intimate contact with the roots. What is the number of bacteria which are there? So what you find, these are the 4 plants which were planted. Okay, the number of bacteria was much more compared to that on the unplanted control. So if there was no plant there, then the number of bacteria was this, but then the number of bacteria greatly increases when the textile dyes have been given or the plants have been watered with the textile dyes. Okay, because certain microbial communities which are already existing there and which are capable of using these dyes as a source of carbon or whatever, they continuously proliferate in these surroundings. Okay, so these are special set of bacteria which you are allowing to proliferate or to multiply in the presence of dyes. Why? Because they can use dyes as a source of carbon, as an additional source of carbon. Okay, so you see 10-fold increase in the microbial count, 5-fold increase, similarly more than 5-fold here, or a 7-fold increase in the microbial count. Okay, the pH, what you see, unplanted control, because you know the textile waste waters are usually alkaline in nature. It's about 8.8. Okay, so so if it is watered with textile wastewater, you have so much, but otherwise you see, because the pH also comes down due to microbial activity, and that is simply because you know they take up certain metal ions which are in the textile effluent, and they also take up proton. They liberate proton because these are pumps which are like proton and metal pumps. So when they take up metal inside, an equivalent amount of proton goes out. And that's the reason why the pH has come down. Okay, similarly, the salt concentrations, etc., you see now are also increased. There is an increase in the conductivity. Okay, so you know, so you have to, because you know some of them, because these bacteria are able to solubilize some metals and some other salts in the soil. Okay, as a result of which the concentration of the free soil could increase, the concentration of the salt in the soil increases, and that's the reason why conductivity increases as a result of microbial activity. So these are all the soil bacteria which you find, which you do not find if the region is unplanted. You know, if you don't have any plants there on the ridge, then the microbial count is very, very small. Now, in the next slide, excuse me. So in the next slide is what is given is the change in the nutrient content and heavy metals in the unplanted and the planted soil with flowering plants watered with the textile wastewater for 30 days. Okay, so now what you see is, for example, if we look at the nitrogen, phosphorus, potassium, organic carbon, this is the control soil which is watered only with normal water. Okay, this is your carbon content, or nitrogen, the phosphorus, the potassium, and the organic carbon content. And if there are no plants there, so this is just the soil, but if there are no plants there, then the number looks something like this. And in the test one, the soil which is now watered with the textile wastewater, okay, you see that the total nitrogen, okay, total nitrogen content has gone low. Similarly, the phosphorus content, if there are no plants, it's 45, but these are the ones, you know, where you have control plants which are watered with the normal water. So there it has gone low, because you know here you see the number is 54, maybe because there are nitrogen fixing bacteria which are associated with a lot of plants. Okay, but as compared to where no plantation has occurred, when you do grow the plants, but then you treat them with the normal water, okay, the phosphorus content is 25, which has gone low from here. Similarly, the content also goes low. That is because all of these metabolites are used for the growth of the plant. Is that point clear to everybody? Please make sure you understand what's happening here.
Yes, ma'am.
Okay, so this particular row is the one where you have the ridge, but then there are no plants there. And here you do have the ridge, you have the plants, but then they are watered with the normal water. On this side, these are the plants which have been watered with the textile waste water. Okay, so the basic control is this. If you have no plants, or both of them can serve as a control, okay, in order for you to understand as to what is happening. And finally, what is shown here are the enzyme activities in the roots of these 4 plants, which are watered with the normal water and textile waste water. Okay, so if you look at the activity, so this is, this is the same plant T. patula. By control, what we mean that on a ridge, these are the 1,000 plants which had been watered only with plain water. The activity of lignin peroxidase is 9.14, but the one which have been watered with the textile waste water, the activity of lignin peroxidase has considerably gone up. Veratrile alcohol oxidase, this is another enzyme which is a part of lignin breakdown. It can also break down textile dyes. Its activity in the control, which is only water, okay, is 73. But then if it is watered with the textile waste waters, then the activity becomes 402. So in all the cases, you see that except this NADH DCIP reductase, there is no difference here. But in all other enzymes, there is a considerable difference here. And the same scenario you find with other kind of plants also, that in all of them, activity of these enzymes has gone up. And plants are known to produce these enzymes, which are lignin degrading. Basically, these are the lignin degrading enzymes. So is this point clear to everybody? Yes or no?
Ma'am, why we have why they are using lignin degrading enzymes? Lignins are like, I think, part of this biomass, like they are produced
Absolutely, absolutely. And
part of this plant, like hemicellulose, cellulose, and lignin, etc.
exactly, exactly. So the lignin, once lignin is made in the plant tissues outside, okay, many of these are also recycled for building up new lignin. So the plants anyway make these enzymes, lignin peroxidase, lactases, which are able to break down these and which are also able to make new lignin. Many of these are enzymes which catalyze both the forward as well as the reverse reaction. So synthesizing new lignin, breaking down existing lignin in the plant cell walls is a continuous activity in the plant. It's like, when you make proteins inside, we can synthesize proteins, so we can also degrade proteins in order to recycle the monomers, which are a part of lignin building activity.
Yes, ma'am. Yes.
Okay, so all of these are enzymes which are normally occurring in the plants, and their function is to synthesize lignin as well as to break down lignin. Now, the only difference is that since, as I showed you the structure, that many of these dyes structurally are very similar to lignin, and that is the reason why the lignin degrading enzymes have been found to be effective on several textile dyes, because they are also aromatic in nature. Okay, so I hope that point is clear to everybody.
Yes, ma'am. Ma'am, this textile, the dyes which are which are present in the effluent, which are which are being ejected from this textile industries. So, if they are all these, only these organic compounds are there, or something like heavy metals, etc., are also there in this effluents.
Well, during the process of textile dyeing, okay, dye is only one part. Apart from that, there are several categories of dyes which are called as complex dyes, okay, in which there is a metal ion also inserted in between. Because iron, copper, like phthalocyanin dyes, they look beautiful blue colors. The shining blue color that you see is a complex dye, phthalocyanin dye, okay, like reactive black 21. If you just Google out the structure of reactive blue 21, there is a metal ion in between, and then it is coordinated to 4 aromatic rings, very much like heme. Okay, now because everybody likes, you all like, we all like to wear colors or fabric in which the dye is very fast. Otherwise you say, what is this, after 1 wash or 2 wash the dye is going away. I'm not going to take such a fabric. We all want fast dyes, but the problem is that these fast dyes, they also stay in nature. They don't get faded by sunlight, they don't get faded by microbial activities, etc. So it becomes a problem. Once these colored effluents, they enter into the water bodies, for example, they retard the photosynthetic activity. So many of the microbial, in the water bodies, there is so much of photosynthetic activity going on. That's not done by plants, that is done by microbes. So those microbes which are carrying out photosynthesis, they depend on certain wavelengths of light because they have chlorophylls, etc., which absorb a certain wavelength. So when you put dyes in the water bodies, okay, much of this sunlight doesn't penetrate into the waters. Dyes are going to block it. So those microbial photosynthesizing systems are greatly affected, and that affects the water aquatic ecosystems because there are no primary producers anymore in that water body. Okay, so one is of course the color aspect of a dye. The second is the toxicity. And it is very well known today that many of these dyes are carcinogenic, they are mutagenic, they can cause cancer. People who are living in areas, where like denim dyeing is a huge industry. Everybody likes to wear denim jeans. But if you see indigo carmine, which is used, it is highly toxic to human beings. And people who are living in those, especially the poor people, who live in these small hutments, etc., near those areas, they drink water which is contaminated with these, and there is high incidence of cancer in these children. Okay, so I hope the point is understood by students as to, why there is so much of thrust on something like textile wastewaters or other pesticides or other kind of things. So is this point clear to everybody?
Yes, ma'am.
So here is a cross section of, anatomy of root. So for example, one of the plants which is T. patula, this is initially, that is before it was watered with the textile waste waters. You see, this is just the cross section of the root. So you will see the outermost layer, the epidermis, and then there is the central, which is the steel part. This is the cortex part, whatever. And this is what happens to it 30 days after it has been watered with the textile waste water. So you see much of the dye is deposited in the root now. It is dark in color. Okay, then B is the second plant, which is A. amylase. This is in the beginning and this is afterwards. So you see the color it's so heavily saturated in the cortex part. And you can also see like all these xylem vessels, etc., are their cell walls, they all have colors in comparison to what you see here. This is the third plant and this is the fourth plant. Okay, so it just indicates that, a lot of this dye, etc., it accumulates in the roots. So therefore, plants are so the take home message is that plants are an exceptionally good, except that they take a lot of time. Then they are seasonal, they don't grow in all the climatic conditions. Then maybe fertilizers and other things will have to be given from outside. So although some places have adopted to this, but it is still not very successful. It's only one of the many ways in which the wastewater, textile wastewaters can be treated. Okay, now in this last slide is given an account, it's from the same paper. that is what you are measuring now is the chlorophyll and the carotenoid content in the plant leaves when they are exposed to textile wastewater. Now, the reason for doing a measure of these plant pigments is that they are also the sites for photosynthesis. Okay, so the question is that if the plants are going to accumulate this pigment, does it affect the chlorophyll content in the leaves? So what you find in T. patula, for example, so by control, what we mean is that, like you see, these are the plants which have been watered with the normal water. So the chlorophyll A content, which was measured, was 9.41, but in the test sample, it was 11.03. Okay, so in fact, the chlorophyll A content has gone up. Chlorophyll B content is nearly the same. It's not statistically significant. Or chlorophyll A plus B, for example, is different, or carotenoids have slightly gone up. Okay, so wherever you see asterisks, that means the numbers are statistically significant. So then if you look at amylase, again, they are all statistically significant. That means in the presence, when the textile wastewater is given to them, the content of these chlorophylls and carotenoid was also found to be increased. Okay, and the photosynthetic apparatus, etc. It's a direct reflection of the metabolic activity of the plant, and therefore it can serve as a useful index. Okay, so the metabolic activity is constantly going on. The idea is that they are not really inhibited because they have been watered with the textile waste water. Doesn't mean that the plants are going to die. In fact, there is an enhancement of this chlorophyll content. So there is an increased metabolic activity. Okay, so I hope you are able to see, all of these and, understand, because the tables would be there for you in order to interpret. So is that clear to everybody? So let's move now. So what are the findings? Is that phytoremediation of the soil containing textile dyes was shown with plants. So, you have all these ornamental plants which have been grown on the ridge beds. So this is a artificially constructed wetland. Okay, because you have the ridges and then you have the furrows, etc. So you have created this wetland onto which the ornamental plants are planted for treatment of textile wastewater. The soil microbial flora was detected. However, no identification was made of the same, because remember, so many bacteria were enumerated, whole numbers. And there was a healthy rhizosphere. Induction in the activity of these oxidoreductive enzymes suggests their probable involvement in the dye removal and phytotransformation. So this mechanism of handling the pollutants by synthesizing enzymes is called as phytotransformation. Okay, so here is an example where you can see phytotransformation has occurred. And anatomical observations of the plant tissues revealed the entry, the accumulation, and further metabolism of the textile dyes, and use of garden ornamental plants looks promising. Okay, so I hope these two papers are clear to everybody as a case study for how, how plants, trees, they can be used successfully for handling some of these environmental problems. Okay, now before we leave, I'm just going to start out with the next topic. So are there any questions so far on whatever we discussed? So any of the students, do you have any questions on whatever? Gopika, are you there?
Yes, ma'am.
And Himanshu, is it clear? Are both the examples clear?
Yes, ma'am.
Okay, so what I'm going to do very quickly is just to get you introduced to. So our quiz is now on Monday. No, our quiz is on Monday now. Yeah. Oh, I see.
14th is the Monday.
14th is Monday, so our quiz is going to be on 14th now. So yeah, so I think let me just continue and introduce this microbial bioremediation to you. Okay, so here is a picture which I just wanted to share with you, and yeah, okay, so this is like from Canada, that what they want to do in their environmental technology wing, they want to bring in plants as a permanent feature while they are trying to treat, for example, the effluent constant, and not only for the textile effluent, but even for other treatment purposes. Plants are finding a way gradually into the normal treatment plants. Okay, so what is proposed to be done? This is from Kawartha Lakes, which is a Canadian environmental development project. Okay, so it's a trust which is still doing a lot of work. And what is anticipated is that when you get these effluents, okay, in which this is a mixed kind of a combined effluent, which is coming in for treatment. So the first step is going to be a pre-treatment in the septic tank, where in pre-treatment, the gravel and the stones and all the particulate matter is allowed to settle down. And then there is an outlet pipe. So it is going to enter into some kind of a wetland, like you see here. Okay, before you give the activated sludge treatment and other things, the water will be now allowed to go through a wetland, into which, for example, a lot of trees you see are going to be planted. Okay, and because of the microbial activity, because of the activities which are resident in the root, etc., there are a lot of these compounds which can now be degraded, which are occurring in this effluent. So it will be passed through large areas of these plants, and then the water is going to be collected. It is going to be pumped now into another kind of a plant setup. So this is a vertical flow cell. Okay, so water is just going to permeate through either the coarse gravel or the sand. First it is through the plants, etc., and then it is collected right at the bottom, because there is a vertical filtration which is occurring, as well as enzymatic activity on account of these plants in this cell. Okay, so at the bottom, because this is a vertical cell, so at the bottom it is going to be collected, and then it is going to pumped into again a wetland, which is a horizontal flow cell, and eventually it'll go like this. Okay, so this is what is planned in many of the Western developing countries. They are also trying to bring in plants into the normal treatment here. Okay, because they see tremendous advantage, and you don't have to, a lot of recovery of nutrients can be done. A lot of things can be treated at extremely low cost in this. So this is like coming in as a technology of tomorrow. Okay, so is this clear to everybody?
Yes, ma'am.
Okay, so let's stop here, and so Saturday we have a class, so we come back and we do microbial bioremediation.